And so I'll put all of these, and I'll try to put the 180s in the back so that I grab those last. So these are the 60-second ones, and I'll put those to the side. And then these are the 30-second ones, and I'll put these in front. So now I'm going to remove all the lids. Alright, so now I'm ready to expose these. So you close the door, you get your timer ready, and so I'll turn it on and start the timer at the same time. And then I will stop the UV at 30 seconds. Alright, so now we'll remove the 30-second ones. And so yeah, that's the 30-second. This one's the 30-second. So now we're ready to continue until they've been exposed for 60 seconds. Alright, so now we'll remove the 60-second ones. Alright, so now all we have are the 180-second ones. And so that would be 3 minutes. So we'll just continue this for another 2 minutes. And I'll probably fast forward through this part. Alright, so it's been 3 minutes. And so now we can remove the 180-second ones and put the lids back on. Alright, so the next step says replace the lid and turn the plate upside down. Repeat for the remaining plates. Stack all 4 plates and use 2 pieces of tape to secure them together. And then place that into an incubator at 35 degrees C and incubate for 2 days. So I will stack them. So this one's Staph epi, this one's E. coli. 30 seconds, Staph epi, E. coli. 60 seconds, Staph epi, E. coli. 180, Staph epi, E. coli. And then I'll just tape these all together and then put them in the incubator, and we'll see what the results are in 2 days. All right, it's been 2 days, and so now we can go over the second experiment that we did for microbial control, which is the UV light, and we exposed our plates to various lengths of time under the UV light. So first is 0 seconds, and so these weren't exposed to UV light at all. So we see that bacteria is growing all across the plate. And so we can reference these to see if our growth was inhibited by the UV light or not. Our next plates were exposed for 30 seconds, and we see that both the E. coli and the Staph epidermidis are growing across the entire plate. The Staph epidermidis looks a little less concentrated than our control, but we do still see a lot of growth. And so for our results, it says record your observations regarding the number of colonies counted for each plate. Record additional data from others in lab. If colonies are uncountable, use TNTC, which is too numerous to count. So now we will look at our plates that were exposed for 60 seconds. So we see lots of individual colonies growing. There is definitely less growth than the control plates, but there's still quite a bit that did survive the 60 seconds of UV light. And you might be able to count this one, but this one would be TNTC. Too many to count. And our last plates are the ones that were exposed for 180 seconds. And you could definitely count these, but we would expect to not see any colonies grow at 180 seconds of exposure to UV light. But I have heard that this happens quite often, and I'm not quite sure why, because these should not be resistant to UV light. But yeah, you would just count each one and then record that in your results.